Hello, welcome to the last part of this segment. We'll be looking at the reporting an experiment. So we've already defined science and we've given us the methods of science. Then we want to look at reporting an experiment. What do you mean by reporting an experiment? Just as it is said, reporting an experiment, that is giving feedback of an experiment a scientist have carried out. If, for instance, a teacher gives you an assignment to carry out a test on a food sample, e.g. bread, to detect or to give the type of food present or food substance present in that bread, how do you report that experiment that you've carried out? How do you give feedback? You don't just write it as if you are writing an essay or summary in English. No, there are patterns that a scientist follows in reporting an experiment. So this is this pattern that we want to study. So reporting an experiment has to do with the patterns that a scientist uses in giving feedback of an experimental procedure in science. So the first pattern is the aim. The aim. What do you stand by the aim? The aim is the reason behind that experiment. At a glance, a layman is supposed to understand what the experiment is all about, starting from the aim. What is the reason behind it? What is the objective of that experiment? For instance, if the assignment given to you, the supposed assignment, your aim could be to test for starch or carbohydrates in a food sample or in a bread sample. That is the aim. So at a glance, I already know what the motive behind the experiment to carry out. At a glance, I already understand the type of experiment that student or that scientist is carrying out or has carried out. So if, for instance, aim now for that assignment could be to test for starch or carbohydrates in a food sample or in a bread sample. That is our aim for this particular study. Then the next one is number 2 is the apparatus. Apparatus is all the equipments, the instruments that the scientist uses in arriving at a conclusion, or all the experiment, all the apparatus, all the equipment, instruments, tools, objects that the scientist uses in carrying out that experiment successfully. For this supposed assignment, our apparatus would be iodine solution, would be a bread sample, we'll also have a pipette. I think basically those three, because of this particular assignment. So the apparatus differs according to what experiment you are carrying out. If, for example, I'm carrying out an experiment on diffusion in liquids, definitely I would have my beaker, I would have my distilled water, I would have my pipette, I would have my crystals of potassium permanganate. But for this particular supposed assignment that the teacher gave the students on test for starch in a bread sample, we'll be needing just the bread sample, our pipette, and then iodine solution. That's for the apparatus. Then after the apparatus, you have the method. The method is the procedures of the experimental setup. What did you do? What are the activities you carried out for that particular experiment? For this supposed one now, I could decide to say I used the pipette to deliver small amounts of the iodine solution from its bottle to the surface of the bread. That's the method. I used the pipette to deliver small amounts of the iodine solution from the bottle of the iodine solution to the surface of the bread in drops, maybe 1 or 2 or 3 drops. That is my method. Then I'll stop there. That's the method that I carried out. Then after the method, you now have the observation. Definitely after carrying out the experiment, there's supposed to be an observation. Even after cooking your food in the house, you add your salt, you add your pepper, Maggi, and allow it to cool. Next time you open it, you must notice that there's a difference. So that's observation, you write it down. So in this our supposed assignment, now will be our observation. The surface of the bread sample turns blue black. That's my observation. After dropping the iodine solution on the bread, then when I look at it, what did I see? I noticed that it turned blue black. Then after the observation, now we have our conclusion. You can call it conclusion, you can call it inference. What will our conclusion now be in this case? Our conclusion would be that starch is present because of the blue-black coloration on the surface of the bread sample. Simple and short. So these are the patterns that the scientists use in reporting or giving a detailed description of how the experiment under science was carried out. The aim has to be stated. A glance at the aim already gives you an idea of what that experimental setup is all about. There, after the aim, the apparatus, all the instruments used in carrying out that experiment successfully has to be listed. Then the method, what are the things you did to make sure that experiment was complete and successful. Then after that, observation. After experiment, there has to be an observation, there has to be a change. Note that down. Then finally, you have your inference or conclusion. We'll be back in a jiffy. Thank you.